Hi, welcome to our OHBM session, Tractometry Peering into the White Matter. I'm Catherine LaBelle, and I'm going to give you just a brief intro to what you're going to hear about today and a little brief intro to tractometry itself. So before we can get to tractometry, what we need to do is get diffusion imaging data from the brain and then use tractography to virtually reconstruct white matter tracts. And you can do this in many different ways, and you're going to hear about some of those methods today. But ultimately, we need to have these white matter tracts so that we can then conduct a tractometry analysis on them. In its simplest definition, tractometry is a quantitative assessment of metrics along or within specific white matter tracts. So you can look at really any metric from sort of classic bread and butter diffusion tensor imaging metrics like fractional anisotropy and mean diffusivity to more advanced diffusion metrics like neurite density index or orientation dispersion index from NADI, or even non-diffusion metrics like myelin water fraction, or really any measure that we have or can generate a brain map of. And then once you have these metrics and these tracts, you can look at different brain alterations that might appear in certain disorders or diseases. You can look at changes following a brain injury. You can look at changes with age, during development or during aging. And you can look at associations between any of these quantitative metrics and things like cognition, behavior, or the environment. So tractometry really is quite a flexible analysis tool to give us information about the white matter tracts and how they relate to any number of different things. So I did a PubMed search for tractometry, and this returned a whopping 18 results over the last 8 years. But of course, tractometry is actually quite a bit more popular than this. We just don't very often call it tractometry. The first sort of type of analysis like this that was introduced was about 2005, when Derek Jones introduced the term pointwise assessment of streamlined tractography attributes, or PASTA. Tractometry itself as a term seemed to come about in 2011 at ISMRM that year. But there are numerous different types of analysis that essentially use these principles. You'll often hear it called something like a long tract analysis. And then there are many studies that don't actually call it anything at all. They'll say they did tractography and then they did quantitative analysis of those tracts, but they won't use the term tractometry itself. Now, we have for you what I think is just a spectacular lineup of speakers, and we've divided the session into sort of two sections. The first section of which will be more on the methodological details. So we have a step-by-step guide to tractometry. We have a talk about clustering and bundling the tracts themselves. We have a talk about some new tractography methods, and then we have a talk about interpreting fiber density metrics. And then in the second section of today, we have talks about using these types of methods to understand different things, so different types of applications. We have applications to high-level cognition, we have a talk about single subject analysis, and then finally we have a talk about using tractometry to study brain development. So we have a number of different ways for you to interact with presenters during this session. Some of the sessions themselves will have tutorials or step-by-step sort of software demos, so please, we encourage you to follow along with those during the session. The presenters will also be available during the presentations to answer questions via the chat function. Most of them will be available, but depending on the time zone, there may be one or two people that aren't available for the entire time. But we do encourage you to ask those questions during the chat, and people will try to answer them as we go. And then following the presentations, we have an hour during which you can ask questions verbally and interact with the presenters then. If you're not watching this during the scheduled broadcast time, you can still type questions into the chat, and our presenters will check back in periodically throughout the week to try to answer those questions as they come up. So we do encourage you to take advantage of these options to try to get the most out of this educational session. So thank you for joining us. I am now going to turn it over to our presenters.
My name is Ariel Rokem, and in this presentation, I will take you through the process of tractometry step by step. Tractometry is a multi-step process in which we start with computational tractography based on the diffusion MRI data. Then we go through a process of segmentation of the tractography results into bundles or tracts. We extract the properties of the tracts along the length of these tracts, or we summarize the properties of the tracts along their length. And then taking those properties, we can do a variety of different things. We compare individuals to each other, populations of individuals to each other, and I'll share a little bit of that as well. And I will take you through a software demo that will demonstrate software that we've written that does all of these things. So let's go through this step by step. Tractometry has several different steps. We generate estimates of the brain connections over the entire white matter. We find major white matter pathways within these estimates. We assess their physical properties, and then we compare across individuals, between different groups, between individuals and groups, and so on. So let's start with computational tractography, which generates the estimates of brain connections that we need. In computational tractography, we take the diffusion MRI data and we estimate the direction of diffusion in every voxel in the brain. For example, zooming in here on a part of the white matter, we look at each voxel in this part of the white matter, and we model the diffusion in this part. And we can use a variety of different models to do that. Here I'm using the DTI model, the diffusion tensor imaging model, to estimate the direction of diffusion in every voxel. You can see in the corpus callosum, there's a lot of right-left diffusion. And here a little bit further out, there's a lot of front to back diffusion. And the principal diffusion direction is used as a cue in this case for tractography. But there are many different methods to do this. For example, we might place a seed in one of these voxels and propagate a streamline. And here, if we place the seed in this voxel, it would follow the principal diffusion direction in every voxel and cross over to the other side. If we put a seed in the neighboring voxel, it might propagate to go back to the back of the brain and following the principal diffusion direction in every voxel along the way. That's called computational tractography, and there are a plethora of methods to do this. Here we've repeated this process several hundreds of thousands of times, and you can see that we get this big bundle of spaghetti in the entire brain. And we can do things like, for example, define a region of interest in the corpus callosum and ask to only get back the streamlines that pass through this region of interest and only in the front of the brain. And that cleans this mess a little bit and gives us only a particular set of connections. And we can use this principle in order to find major anatomical tracts or major white matter pathways within these estimates. And we know from many years of anatomical studies that the white matter is made out of different tracts, and we call those sometimes bundles because in the streamlines, those are not anatomical tracts. Those are models of the tracts based on computational tractography. And those aim to approximate these anatomical connections that every individual brain has. And we rely on an idea of virtual dissection. Given a bunch of streamlines as here, we might say, well, if we're looking for this particular tract, say the arcuate fasciculus or some other tract here, we might say, we'd like it to pass through this waypoint ROI here in the front of the brain and this other waypoint ROI. And streamlines that have this trajectory, we say that those belong to a particular tract in the brain. Here, for example, we've placed two waypoint ROIs in a way that isolates streamlines that belong to the corticospinal tract. Here, placing a different set of waypoint ROIs, we can find streamlines that belong to the arcuate fasciculus, the left and the right, or in this case, to the inferior fronto-occipital fasciculus by placing another set of waypoint ROIs. Using this approach, we can find a variety of different tracts. Another alternative approach to this uses match between streamlines in streamline space to an atlas. For example, here's an atlas that Frankie developed from the data in the Human Connectome project and published. And here I'm using a method that Eleftherios Garofalidis developed called RICO bundles. And this method tries to find streamlines whose trajectory matches more or less the trajectory of the streamlines that are in the bundle. So here the match is done instead of by looking for waypoint ROIs along the length of the streamlines and the fact that these streamlines pass through these waypoint ROIs. Instead, here we look for something that has a trajectory that resembles the trajectory of the atlas. For example, here we identified the optic radiation in a single individual subject on the left and on the right based on this atlas bundle. The next thing that we'd like to do is to assess the physical properties of the bundles along their lengths. This idea called PASTA, pointwise assessment of streamline tractography attributes. I wish that I could say that I came up with this acronym, but it was invented by Derek Jones and colleagues in 2005 to describe the following process. Let's say that we have a bundle and we've measured diffusion along the length of this bundle as well, and we've assessed and modeled and estimated, for example, the fractional anisotropy along the length of the bundle. If we divide the bundle up into nodes, we can do pointwise assessment of the FA at different locations in the bundle. So we look at the FA around this first node, and we put that as the first point in this curve, and then the next point, and put that as the next point, and so on and so forth. And that gives us a full set of points. In this case, we've assessed FA in 100 different nodes along the length of the streamline, and that gives us what we call a tract profile or a bundle profile of 100 different values of the fractional anisotropy, and we can do that for every individual bundle in the brain. Now we've written software. John Kruper, whose name is mentioned down here, and whose picture I will show later, wrote software called PyAFQ. This is based on software that Jason Yateman and colleagues developed in 2000 and published in 2012. That was developed in MATLAB, and now we've updated this to be a fully open source Python software library. And I'm going to show you a little demo with code that shows how we use PyAFQ to do these various steps. So if you use this, you can also follow along. If you go to this URL here, tinyurl.com/pyafq-demo, that will take you into something that looks a lot like this. This is a collaboratory Jupyter notebook. And if you click on File and you click on Save a Copy in Drive, then it will create another copy of this notebook for you. And this is a copy that you can run on your end. So this notebook, you can run it by hitting Shift Return, and that will run the first cell here. I'm going to clear the outputs. The first cell at the top here goes and installs the PyAFQ software for you. And then while this is installing, I'll walk you through the code that will happen afterwards. Here I'm importing the main API of the software from the AFQ library that will be installed into this collaboratory notebook that you can run. I'm importing a little data module that you can use in order to get some data. When I hit organize Stanford data and execute this, it will go and it will get some data that I collected a few years ago when I was a postdoc at Stanford, and it will organize that data inside of your file system in this machine. So if you open up the file system here and you click on the root folder here, that's where the data will show up once it's the software has been installed. Like in a good cooking show, I will now go to another instance of this where I've already completed these steps. Here you can see that after running the software, there's some data. It's organized in here in a BIDS compatible format for you. So the input data was processed with VistaSoft, and so in here you'll see some DWI data that's organized according to a BIDS-like format, and that's downloaded when you execute this organized Stanford data cell. Now I'm going to import a few definitions that we need in order to generate the results of the AFQ processing. Here is the main object that executes the processing. We point it to a particular path. In this case, we're pointing it to this Stanford Hardy dataset path, and it expects to have a BIDS compatible, pre-processed BIDS compatible diffusion MRI dataset in that location, and it will look under the VistaSoft folder within here, within the derivatives folder. It will look under the VistaSoft folder. It treats VistaSoft as the pipeline that ran the pre-processing, but this can be other software as well. For example, we use QSI prep in order to do a lot of the pre-processing of the data that we're currently doing, and we're working on developing DMRI prep as well as a software that will do the pre-processing that precedes the processing that we're doing here. Now you can give AFQ instructions about how you'd like to do the processing. In this case, we're giving some instructions about the tracking parameters, how many seeds to place within every voxel of the white matter that we're designating as seed voxels. Here we're using as a seed mask only the regions of interest, those waypoint ROIs that I described to you before. We're going to do deterministic tracking, and our model of the ODF is the DTI model that I showed you before, but these are things that you can configure. Bundle info tells us what bundles we'd like to find, and here are some instructions about how to map between the template, the Atlas template, where the waypoint ROIs are, and the data of this individual subject, and then how to plot the results. So this, when I run this cell, it will define these things. It won't run any of it won't execute any of the computations yet. Instead, when I ask for particular properties of this MyAFQ object, for example, I'm interested in getting the DTI FA, the FA map out of this, then it will execute the fitting of the model. And this, on your end, the first time you run this might take a little time until the model is fit. It won't happen as fast, but after it fit that, it will cache the results. And so in the AFQ folder, under the derivatives folder, in the Stanford Hardy folder that I show you here, in this subject and session, you'll see that it's generated a variety of derivatives. For example, it's generated this DTI-FA map, and this is the thing that it's loading here. Use matplotlib in order to plot a map of the FA. So this is the FA, and you can look at that. Next, in the next cell, what I'll do is I will ask to visualize the bundles. Again, if you're running this for the first time, this could take a little time. This step in particular may take about 10 to 20 minutes to run, and that's because along the way it's generating the streamlines, doing the tractography, and that can take some time, and also it does the bundle segmentation and all the process of finding which streamlines pass through the waypoint ROIs for the target bundle. In this case, as I said, the arcuate fasciculus that we're targeting in this case. So this can take a little while here. So maybe I'll stop the recording here, and then I will come back to the recording once it's done. And then I'll show you what we got here. When you run this on your end, this might take a little while. Like I said, the entire notebook takes about 20 minutes to run from top to bottom. So I'll be right back. Okay, I'm back. So the processing here finished, and now we can execute the next thing, which is to plot the track profiles, calculates the means and confidence intervals for every track across individuals. In this case, we only have one individual. Now we can read this track profiles variable here will hold for us the name of the file where the track profiles have been saved as a as a comma separated values file, read that into using pandas into a data frame, and then we can do all kinds of manipulations. For example, we can find the part of these track profiles that corresponds to the arcuate, and within those track profiles to find the DTIFA, and then we can plot them. And so here we have a plot, for example, of the FA in the arcuate, the left arcuate and the right arcuate that are labeled. And along the way, we also generated a variety of different output files. One that I like to point to in particular is this HTML file. If you download that HTML file onto your own machine, like this, and then drop that into a browser window, you'll see another kind of visualization of the results of the tractography and the bundle segmentation. These are the arcuate left and arcuate right that were found by the algorithm. You'll see that they're not very big in this case. That's because usually when you want to do this, you want to maybe track a little bit more than you want to probably track more than the 1 seed per voxel that I generated here for the sake of saving time. But this allows you then to explore the results in this interactive format. Okay, that concludes the demo of PyFQ. So after you've done that, you might consider, well, I showed you along the way that I made all kinds of choices for how to do this. And you might be a little bit worried that there are too many options. This is something that others have highlighted recently in this paper. With Phoenix-Nesser et al. Gave a variety of different research teams the same fMRI data set and asked them to test a variety of different hypotheses about this data set. And they saw that different choices made at different steps about it. For example, spatial smoothing, statistical modeling, or thresholding affected the inferences that different research groups made. And you look at this, and this is a flowchart that John prepared to explain all the different choices that you can make along the way when you're using PyFQ, choices about the tractography algorithms that we use, and there are many options there. They're the kinds of registration algorithms that we use to match the individual subject's data to the template. The choices we make in terms of bundle recognition, whether it be using the waypoint ROIs or using the Reco bundles algorithm that I described before. All of these require making a variety of different choices, and you might worry that that makes these results less replicable, less robust, using this terminology that Kirsty Whitaker from the Turing Institute defined, where if you use the same data and the same analysis, you might think that the results are reproducible. But even if you use the same data and different analysis, you might want to still get the same results. So different choices here, you might want them to lead to the same results, making your analysis more robust. And of course, we all like these results eventually to also be generalizable, replicable on different data sets and using different data sets with different forms of analysis. So what we asked is whether these choices, whether the analysis is robust to these choices. So here we're comparing two different algorithms that were used for tractography. Here we're zooming in on the superior longitudinal fasciculus and the arcuate fasciculus. And what we see is that using different models of the ODFs of how to determine the directions of tractography in the different voxels can have pretty profound consequences on the way that the tracks appear in this representation. But if we look at the robustness of the track profiles, looking at the correlation within individual between the track profile generated with this algorithm and the track profile generated with this algorithm, in some places, in some bundles, here are different bundles that we do this for. In some bundles, the robustness is relatively low, but in some bundles, the robustness can actually be quite high. And this is true both at the level of the single individual, but also at the level of comparison, taking the mean across the tracks and comparing across individuals. So this is cross individual correlation between the DKI and CSD derived average track profile. You can see this is both true for both FA and to a lesser degree also true for the mean diffusivity ND. Another example of this is that you can use two different algorithms that I described to you before to find the tracks. So here we're using the waypoint ROIs to find the inferior longitudinal fasciculus, and here we've used record bundles in a particular configuration to find the inferior longitudinal fasciculus. And again, you see that profile robustness in some cases is very, very poor. For example, these ILFs look very different, and indeed the correlation between the tracks within individual are quite low. But in other cases, actually using algorithms that are very, very different, you might find a pretty good match across these two different methods. And this is true also for the subject level robustness, even more so for subject level robustness. So that's encouraging. The next thing that we'd like to do is to compare across individuals between different groups and between individuals and groups and so on and so forth. Take and do statistical analysis with the tractometry data. So one approach is to compare directly the track profiles to each other. In this case, this is data from a group of patients with amyotrophic lateral sclerosis, or ALS, in blue. This is the FA and their corticospinal tract, and a group of matched controls here in their FA is in red. And you can look and see that in the corticospinal tract, in these individuals, there is a point-by-point difference. And you can do a statistical test for every one of these differences. And that's a way to assess whether there are differences between these groups. And indeed, in this case, there is a pretty dramatic and statistically significant difference. But there are more sophisticated ways of doing this, more elaborate ways. One example that Branch Chandio and in Eleftherios Garifalidis' group at Indiana University has developed is called bundle analytics. And bundle analytics is a different way than the one that I showed you before to do the pointwise assessment of the track profiles. And it uses linear mixed models to do the assessment of the statistical significance. And this is implemented in software called DiPy, diffusion imaging in Python. This is open source software for analysis of diffusion MRI that I collaborate on developing, and that we rely on very, very heavily in development of the Python AFQ that I mentioned before. So shout out to that software. So that's one approach. You take two groups and you compare them to each other and you assess the statistical significance. Another complementary, different approach is a prediction approach where, for example, you might look at different individuals here. Maybe this is the age of individual number one, the age of individual number two. And you might want to build a statistical model or a machine learning model that predicts the age of every individual based on the diffusion MRI data. So this focuses on a prediction rather than an assessment of a difference. And so you could fit a statistical model, in this case, linear model y equals x, and here the x, every row in this x matrix contains all of the diffusion properties along the length of all the tracks within each individual. So each row here is a single individual, and so these are all of the pasta values for individual number one, and then under that there's another row here with all the pasta values for individual number two, and so on and so forth. And using a variety of methods, we can fit a linear model that solves for the beta coefficients that go into the prediction. Using this approach, Adam Ritchie Halford, who's in my group, developed a set of algorithms that rely on the fact that these properties are grouped according to the different bundles. And these algorithms work very well. For example, they accurately predict participant age here in the Cambridge Center for Aging and Neuroscience CAM-CAM data set. This data set contains almost 700 individuals, and this kind of linear model predicts their age here in a held out cross-validation sense, predicts very accurately age of individual subjects. And then we can go and look at the beta coefficients that we fit here across different bundles in the brain and see how different parts of the brain play into these age differences between individuals. Finally, one more thing that I'd like to show you is a tool that we developed that puts some of these things together. This is a tool called FQ Browser that takes the results of the Tractometry pipeline that I described before and allows you to build this kind of interactive web-based browser that visualizes the results of a diffusion MRI study, a tractometry analysis of a diffusion MRI study. Here, we're exploring the corticospinal tract in the ALS patient group and controls from the data that I showed you just before from a paper from Anastasia et al. From 2017. And the video that I'm showing here demonstrates how we explore the data, showing different parts, sorting based on different attributes, and so on and so forth. Okay, so to wrap up here, I'd like to thank my collaborators on this work. I'd like in particular to highlight John Kruper, who's the main developer of PyFQ, Adam Ritchie-Halford, who did the work that I showed about the prediction approach to tractometry. I'd like to again thank Eleftherios Garifalidis, and these are some members of the DiPy team at Indiana University, in particular Ben-Shachar, who developed the Bundle Analytics Buan tool, and then a lot of other contributors, as you can see here. In time, we've grown. The DiPy project has grown in terms of the number of contributors, and we welcome contributions from anyone. And I'd like to thank the sources of funding and support for the work that we do, and I welcome you to check out our work on this website. Thank you.
Hello, everyone. I'm Anastasia Yendiki, and today I'll be talking about ways in which we can use tractography to extract anatomically meaningful bundles that we can then compare across individuals. There are two general strategies for using tractography to extract white matter bundles. One is the atlas-based approach, where we use prior information on major bundles that have been defined in anatomic literature, such as the uncinate fasciculus, the cingulum bundle, et cetera, et cetera. And this approach allows us to reconstruct these same bundles in a bunch of subjects automatically. The alternative is the data-driven approach, where we do not have any a priori tracts of interest. Instead, we run tractography on the whole brain, and then we group the streamlines into tract clusters based on their similarity. This allows us to perform exploratory studies of whole brain tractography data. So let's first look at the atlas-based approach. The vast majority of methods that have been proposed for automated reconstruction of major white matter bundles are variations of what we call the multi-ROI method. The idea here is that each major white matter pathway can be defined by 2 to 3 ROIs that it goes through, and perhaps a couple of ROIs that it doesn't go through. And the steps in a multi-ROI method are to first run tractography, either seeding in the whole brain or part of the brain. Then to draw regions of interest in each individual subject manually. And then hope that there are going to be some tracks that connect these ROIs that define the specific bundle. There are two drawbacks here. One is that step 2, the manual labeling, is time consuming and it requires extensive neuroanatomical expertise. And also that step 3 is not robust. So we are not guaranteed, even after doing all this work, that we will find tracks that go through all the ROIs. The automated flavor of the multi-ROI method simply takes the ROIs from an atlas instead of having to draw them manually. And again, tractography is run on the whole brain. Now the ROIs are mapped from a template to the individual. And as before, we hope that we find tracts in the individual that connect all these ROIs. This approach removes one of the drawbacks, which is the time-consuming nature of drawing the ROIs manually, but it doesn't solve the other drawback, which is the robustness issue. So whether we'll actually find tracks that connect the ROIs that we want depends greatly on the data quality of each individual subject's data and the difficulty of the specific bundle that we're interested in. To address these issues, we introduced an alternative to the multi-ROI approach to automated tractography. It's called Tracula, or tracks constrained by underlying anatomy. And instead of running tractography and then filtering it through some deterministic post-hoc ROIs, the algorithm learns the prior probabilities of the anatomical neighborhood of the bundle of interest from a set of training data, and then uses these priors in the tractography step itself. This is done within a Bayesian framework where the likelihood term fits the shape of the bundle to the orientation vectors from the subject's diffusion data, and the prior term fits the shape of the bundle to our prior knowledge on the anatomical neighbors of the bundle. The set of potential neighbors are the labels of the free server segmentation from the subject zone T1, and we have a bunch of manually annotated bundles that are used to compute the prior probability that any given label is the neighbor of our bundle of interest in the lateral, medial, rostral, caudal, etc. direction as a function of where we are along the bundle. This idea is stolen from anatomists. So in the anatomic literature, axon bundles are not described in terms of MNI or some other template coordinates or ROIs that they go through. Instead, they are described in terms of the anatomical structures that they go through or next to. In this example of a fiber bundle that's described in the Schmamann and Pandya book, we learned that this bundle goes below and medial to the claustrum, then travels until the most caudal level of the LGN. Then part of it goes medially and terminates in the thalamus. Another part goes over the rostral and middle sectors of the LGN, and so on and so forth. So the bundles are defined based on their anatomical neighbors. And doing it that way makes it easier to translate this information across individuals and even across species. As an aside, another place where this idea is used is actually the free server segmentation. It calls these Markov random field priors, and they encode how likely each segmentation label is to occur lateral, medial, rostral, caudal, etc., to any other segmentation label. These priors are computed from some manually labeled brains. So Tracula essentially does what free server does, but instead of doing voxel-wise segmentation, it uses this idea and applies it to tractography. In the first version of Tracula, we could reconstruct 18 bundles that covered only 18% of the white matter. In the latest version, which is being released with free server 7.2, we can reconstruct 42 bundles that cover 82% of the white matter. This increased coverage comes not only from having more bundles, but also from having fuller definitions of these bundles from higher quality data. So in our old version, our training data were manually annotated bundles that came from conventional scans of that era. This is an example of how much of the SLF we could get from one of those training scans. In the new version, the training data are manually annotated bundles from the MGH HCP data from our Connectome scanner. And this allows us to get much more complete manually annotated bundles, as seen in this example that shows nice delineations of the SLF 1, 2, and 3. Our goal is to take advantage of these much better models of white matter bundles that we can get from the Connectome data to improve the automated reconstruction in conventional data, so that you don't actually have to scan your subjects on a Connectome scanner to take advantage of these data from the Connectome scanner. And more specifically, we looked at using the training data from the Bmax equals 10,512 direction scans of these Connectome subjects and doing the manual annotation on these scans, and then training Tracula to reconstruct the same bundles automatically using only the B equals 1,000 scan of the same subjects with 64 directions or even just 32 directions. The good news is that both with the Tracula and with the multi-ROI approach, we can achieve pretty high sensitivity in the B equals 1,000 data with respect to the B equals 10,000 data. This means that for this task of reconstructing major white matter bundles, it's not actually critical to have very high quality data. However, to achieve a certain sensitivity level, different methods have to tolerate different reconstruction errors. So to achieve a sensitivity level of 89% in this lower quality B equals 1,000 data, Tracula, when run on the 64 direction data, has a reconstruction error of a couple of voxels, or 3.5 millimeters, which then increases to about 20, by about 20% when we go from 64 directions down to 32 directions. For the multi-ROI method, however, in order to achieve that same sensitivity, it had to have twice as high reconstruction error in the 64 direction data and about 3 times as high in the 32 direction data. And that's because the multi-ROI method is largely dependent on the data quality of the individual subject to be able to find the tracks that connect the different ROIs, whereas Tracula takes advantage of these more informative priors that we extract from the high quality connectome data, and uses the information on the anatomical neighborhood of the tracks from these priors. And so it suffers less from a potential drop in the quality of the test data. Another direct benefit of the Bayesian framework that we use in Tracula is that it can be readily extended to model longitudinal data. In a longitudinal study, the goal is to reconstruct a white matter bundle consistently across all time points at which a subject is scanned. And this can be challenging when the tractography is performed on the images from each time point separately. We may end up with part of a bundle missing at different time points, depending on the different levels of noise, degeneration, et cetera, at each time point. And this means that we would be comparing apples and oranges across the different time points. And this is particularly a problem for establishing point-by-point correspondence across the time points to do the kinds of a long tract analysis that we're talking about in this course. With Tracula, it is straightforward to extend the likelihood and the prior terms in the Bayesian framework so that they operate on the data from all the time points at once instead of just one time point. And this allows us to reconstruct the bundles jointly, given the data from all the time points of the given subject. And this ensures point-by-point correspondence along the tract. And as a result, as we've shown in our 2016 paper, this increases sensitivity for along the tract longitudinal analyses without sacrificing retest reliability. Last but not least, we have a version of these tools that works for infant brains, and it's called Traculina, or tracts constrained by underlying infant anatomy. It is trained on data from both full-term and premature neonates. One thing that people often ask is, does it matter if the anatomical segmentation from FreeSurfer has errors? And in this infant work, we showed that we do not actually incur any significant drop in accuracy if we perturb the boundaries of the segmentation labels or the inter-subject registration matrix to simulate segmentation or registration errors. The reason for this robustness is that, unlike, let's say, in a cortical thickness analysis where you need sub-millimeter level accuracy of the tissue boundaries, here we only use the segmentation to learn relative positions of structures. So which structure am I medial or lateral to? And this is robust to having small errors in the boundaries of the structure. So we do not need a super accurate segmentation. Now let's look at the other approach, the data-driven approach, where we do not have any a priori tracks of interest, but instead perform clustering on whole brain tractography streamlines. There are a couple of main approaches for doing clustering. One is flat clustering, where we choose the total number of clusters that we want, and the streamlines are grouped into that many clusters in one shot. The goal is always with clustering to maximize similarity within each cluster and minimize it between clusters. Another approach to clustering is what we call hierarchical clustering, where we iteratively split the whole brain streamlines based on similarity, first into 2 groups, and then each group into 2 subgroups, and so on and so forth. This gives a hierarchical tree of track clusters that start as big groups, like, let's say, all the left hemispheric connections or the right hemispheric connections and the interhemispheric connections. And then as we go down the branches of the tree, we get into more and more localized connections. And it is also possible to do this in reverse order, where we start with each streamline being its own cluster and then merge them iteratively until we get from the leaves all the way up to the base of the tree. Regardless of which of these clustering approaches we use, we have to choose a similarity metric. So we have to define what it means for two streamlines to be similar or not. The usual similarity metrics are based on the Euclidean distance between the points of the streamlines. So two streamlines are grouped together if they are close to each other in space. However, we can imagine a scenario where two tracks are close to each other in space, but actually go through very distinct regions anatomically. Because of this, and because we like the idea of defining bundles based on their anatomical neighbors, like anatomists do, we came up with a different similarity metric. So instead of describing each streamline by the coordinates of its points, we describe it by the histogram of its anatomical neighbors in different directions, in the medial, lateral, caudal, rostral, etc. direction. And again, the neighbors come from the subject's own free surface segmentation. We then group streamlines together if these histograms of their neighbors are similar, and not based on how close the streamlines are to one another in space. A benefit of describing tract clusters based on these histograms of the neighboring anatomical structures is that these histograms are computed in native space, and then they can be compared across different subjects without having to co-register the subjects in a common space. So after we cluster the tracks based on this anatomical similarity within subject, we can then also use this anatomical similarity to identify corresponding tract clusters across subjects, again by comparing the histogram of the anatomical neighbors of each of the clusters. And we've shown that in comparison to the conventional Euclidean distance-based similarity metric, this anatomical similarity metric has several advantages. First of all, when it is used for within subject tract clustering, it yields clusters that are 20% more similar to manually labeled anatomically defined bundles, even though it does not use any training data at all on any a priori defined bundles. So there are no priors here, and we only use the subject's own tractography data. It yields more realistic anatomically meaningful clusters only because it uses the same philosophy as anatomists, defining the clusters based on their anatomical neighbors. And when we also use this approach to find corresponding clusters across subjects, it leads to 38% more similar clusters between individuals ranging in age all the way from 9 to 90 years old when compared to a conventional distance-based similarity metric. Once we have this correspondence of cluster to cluster across subjects, then we can perform a long tract analysis on these clusters that we have extracted from the data-driven approach, just like we would from the bundles that we would extract with the atlas-based approach. So in both cases, we want to compare microstructural measures point by point along each bundle across subjects, across individuals, and across groups. This is not a new concept. It's actually almost as old as tractography itself. But instead of tractometry, it used to be called something else. Derek Jones introduced the term PASTA, or point-wise assessment of streamline tractography attributes, in this 2005 paper. All of the elements, computing the microstructural measures at consecutive cross-sections of the bundle, performing statistics on them, and then mapping the results back onto the 3D shape of the bundle, were there. And then several other groups refined the ways to project the the microstructural measures from the volume onto this 1D profile. And I think that PASTA is a brilliant acronym for describing analyses of spaghetti-like data, so I'm going to go with that instead of tractometry, but keep in mind that we're talking about the same thing. So briefly, how do we implement PASTA-style analyses in Tracula? We build on the widely used and validated free surface tools for statistical analyses. So the same tools that you would use to do stats either in the volume in 3D or on the surface in 2D, you can also use for PASTA, which is simply a special case where we only have one dimension, the along track dimension. For those who have performed stats in FreeSurfer, let's say for cortical thickness or subcortical volumes, you know that any number of discrete or continuous covariates can be defined in what we call an FSGD, or free surfer group descriptor file. And these same FSGD files that you may have used to analyze cortical thickness, etc., you can also use for PASTA style along tract analyses on the different bundles that you would get from Tracula. The usual free surfer commands apply here. So MRI GLM fit performs uncorrected analyses, and then MRI GLM fit -sim performs simulation-based cluster-wise correction for multiple comparisons. Here, I'd like to thank my main co-conspirators from the free surfer development team, as well as the tract annotation team that built the new 42 bundle tract atlas that we're about to release this month as part of free surfer 7.2. The team was led by Chiara Maffei, and they're all shown here with their pet tracts. So to recap, we looked at two different strategies for defining anatomically meaningful bundles: the atlas-based approach that allows us to reconstruct predefined tracts of interest, and the data-driven approach that allows us to perform exploratory studies on whole brain tractography data. We saw that specialized tools exist for analyzing longitudinal and/or infant data. And we also saw that a long tract stats can be performed with all the usual free surfer tools as a special 1D case of the usual surface-based or volume-based analyses. Some of the features that I mentioned in this talk, and particularly the new 42 bundle package, will be available in free surfer 7.2, which is set to be released during the month of May. So check closer to the OHBM educational course dates, both for updated tutorials and documentations at the usual spot on the free surfer wiki, as well as some special Easter eggs from a new tract atlas on the new website that we are migrating to, dmri.mgh.harvard.edu. Thank you very much for your attention.
Hi, I'm Frank to talk about new tractography modalities for tractometry analysis. Here's a brief outline. I will first begin with a recap about tractometry and show you how to do it in DSS Studio. Then I will introduce differential tractography and correlational tractography. Other talks has talked about what tractometry analysis is. To summarize here, the first step is to map white matter tracts. So we could use either ROI-based segmentation or either automatic tractography to map the target pathways. Then we'll use those tracks to sample diffusion metrics and get profiles or get any kind of index statistics and try to find a difference. So in overall, tractometry is a two-step process. First, to map fiber tracks, and the second one is to find individual difference or either group difference. So here's a short video showing how we could do this in DSS Studio. We'll begin with Nifty file and bval bback file. You can get those files from FSL's top-up Eddy, or either get it from DCM to MRI. First, we will convert Nifty file using step T1. This will match Nifty file with bval and bback. So open it up, and DSS Studio will show you on the match result. Just click OK. Then you generate a new file and with src.gz. Once you get this file, you can double click here to open it, or either open it in step T2 reconstruction. So open it up. DSS Studio will show a mask. This brain mask will cover the brain region just to check whether it covers well. On the top, there will be buttons. On the button, there's a slide bar. You can check each of the slide. If the mask is not good, you can modify it here. Then we can select reconstruction method, DTI, GQI, QSDR. Here we show GQI reconstruction. Click on reconstruction, then you DSS Studio generate a fit file. We can double click it to open it up. Here we will go to show how to do automatic tractography. On the right, there is tracking parameter. You can click on the button in the middle, then we can select different targets. Say, for example, here, arcuate fasciculars. Click fiber tracking, and you will see DSS Studio automatically map this pathway. We can also set a different pathway, adding them to the list, track each of them. So this would be the first step to do tractometry. We need to map all the pathway we are interested in. Now we have all tracks mapped here. I assign different color, and you can see whether the mapping is accurate. Then we'll move to the next step, which is the tractometry analysis. To give a simple example here, I will use IFOF, 3 different branch of it, and to show how we could get the whole track statistic or track profile, or either the anisotropy or metrics along the tracks. So I just show different color here to let you see 3 different branch, IFOF 1, 2, 3. The first step is to get a statistic of the whole tracks. You can see I could click on the statistic button and copy to the clipboard, and I can paste it to Excel file. And you will see on the first few metrics, there are shake measures, and followed by diffusion metrics like anisotropy or diffusivity. In addition to whole track statistics, I can also get track profile like FQ. Here I switch to the Y directions because IFOF is going from anterior to posterior. We could get the value along the Y axis. So you can see here 3 different curves representing 3 branches of IFOF. You can look at different metrics, FA, mean diffusivity, SO diffusivity, and all kinds of different measurements. I can also look at the fiber orientation. In this way, DSS Studio stretch each track to the same length and then get the profile instead of just looking at the Y direction. So another way to look at those measures is to check the long track index. So here I switch to the local index. I can visualize FA along the track. I can also visualize all different measures and change how the color profile showing an anisotropy along IFOF. So in practice, when we work out tractometry study in DSS Studio, we always start with just one simple case on GUID, and we recursively checking the methods, parameters to make sure that it's accurate and the result is good before we move forward to process all subjects data, either using GUID or command line interface. So in GUID interface, the user could set up multiple files. There's no need to write a script, and it's suitable for a simple processing flow. And of course, for command line, it allows for more complicated processing flow, and is the only choice for job scheduling on clusters. Well, if the tractometry show good results, then probably we don't need more complicated methods. However, in reality, there are limitations. So I'm showing an example here. There's a brain tumor patient with a brain tumor here, and we are mapping like the restricted diffusion around the tumor, see where the tumor cells are infiltrating into tracts. And you can see here's a lot of time the findings are really focal, and just involve a segment or just a branch. And tractometry analysis, sometimes you could average out those effects or just missing those tiny foci here. So what could we do? Remember, tractometry is a two-step process. First step is to map fiber pathway. In this step, meaningful finding may not be mapped, may not be covered. And in the second step, we do those profiling, statistic or data reduction, meaningful results may be removed or averaged. So I came up with a solution that is, why not combining these two steps into just one? We could track differences directly in the fiber tracking algorithm. Instead of a two-step process, we can have a simple one-step process to map out the location of the meaningful finding. And this gives us a new modality called differential tractography. How does it work? So in conventional tractography, we have termination criteria. Say, for example, the anisotropy value. If the FA value lower than a threshold, then the tracking will stop there. Or either there's a large turning angle, the track will not continue tracking. So those criteria define where the location and coverage of the tractography. For differential tractography, it inherited the same framework and just adding a simple new criterion, that is the difference of the matrix we are interested in. Say, for example, if we want to look at decrease of FA, we could say, okay, here we have one new criteria is to track if the FA decrease that's more than 10%. Then the tracking will map all the location with track that has FA drop more than 10%. And this gives us a differential tractography. We'll use one TBI patient data to show you how to get differential tractography. The data we have, including a baseline scan and a follow-up scan 1 year later of the same subject. The first step we are going to do is using step T1 to convert NIPT file into SRC file. DSS Studio will make sure that the DWI match well with their B table. And once we have those SRC file, we'll open the baseline scan in step T2. Just as the same as what we did here, but here we will click compare SRC button and select the follow-up scan. And in this way, DSS Studio will compare baseline and follow-up to calculate voxel-wise difference. This step will take quite a while, but we fast forward. Once the analysis complete, we'll get a fit file that we could open it in step T3, fiber tracking, to get differential tractography. So the file here, we could double click here to open it, or either use step T1, set the fit file. Now DSS Studio will show the interface or the 3D. First, we will get MP range to look at the lesion location. This will help us to check where the finding is meaningful. Then before we go for differential tractography, we will first get a whole brain conventional tractography just to track the quality of data. Sometimes the B table may not be correct, or maybe there are artifacts or any kind of signal issue. And here we could see, well, the whole brain tractography looks okay. Then we can turn on differential tractography on the right. Here in the matrix, we will look at decrease of FA. So 0.1 here means we look at difference at least more than 10%. And click on fiber tracking. At first, DSS Studio didn't show any findings here because we only have very few seeding points. So just add more seed points and run fiber tracking again. Now you will see there's a large difference that's showing where those tracks has F rate decrease more than 10%. So this gives us a differential tractography. Well, you may be wondering whether the result is reliable, because we can always find a difference between any two scans. So in the original paper, we include a framework to quantify the reliability using false discovery rate. So for details, you can check the original paper. And on the DSS Studio website, there are tutorials to guide you how to calculate the FDR. So how about group studies? Say not just individual, just single subjects. We have two groups of study population. And how about regression model, covariates? Sometimes we need to regress our age effect, gender effect, or any other effect. How could we use the same paradigms here? So one way we could do is, well, instead of tracking difference, we could track correlation by adding a t-statistic threshold as a termination criteria. So just the same paradigm, but instead of tracking difference, here we track correlation. Last year, we have used correlational tractography to study stroke patients and to visualize tracks that are positive correlated or negative correlated with language functions. Here I'm going to show you how to do this in DSS Studio. We'll prepare a file that's db.fib.gz. This one is created from step C1 and step C2. So the file aggregated 60 subjects data into just one file to simplify the process. We also need another file that's the subject's demographic. You see here, we have gender, BMI, age. We could also have missing value labeled by 9999. So this one, we could also use comma separated value. Either file will work out as demographic. You can use a text editor or any kind of editor to change it, to edit them. Once we have this file, we open up the zip file in step C3 to get correlational tractography analysis. Open it up, and DSS Studio will show interface that you can load the demographics. Here we could open up the comma separated value or the space separated value. You check whether the value match well with the subjects. On the lower part, we will select covariates. Here we have 3 selections. Check them to include in the model. And on the right-hand side, we could select which variable we are going to study. So here I'm selecting the BMI to look at which tracks are correlated with BMI those subjects. There are parameters at the bottom. You could check where you want to have FDR control or either just report FDR values. There are also T thresholds that determine whether strong correlation or weak correlation, and also the length threshold to eliminate fragmented results. You could also set that study region, exclude cerebellum, or either you can assign a specific target just to look at which tracks and see whether they are correlated with the variable. Here we also can set that the subset of subjects. For example, here we could say, okay, I want to just look at studies, those subjects with age older than 25 years old. And you can see here, DSS Studio will highlight those subjects and ignore other subjects in the analysis. So this function allows us to say, okay, I want to just look at a subset of subjects. And instead of just looking at all of them, we can also exclude missing values here. So if there are missing value, we can also exclude there from the analysis. You can set any kind of combination of variable just to define your cohort in the study. Once ready, you can click Run Tractometry Analysis to get correlational tractography running. So on the right-hand side, you will see several plots, one for positive correlation. On the right, there is negative correlation. They will be analyzed separately. You'll see two lines, one for permuted results, another one for non-permuted results. So the reason here is DSS Studio will use permutation test to check the reliability of the results and calculate the false discovery rate. So as you can see here, the analysis is running, and DSS Studio keep permuting the demographics to see if the result is reliable. So here we just have 6%. We wait until it goes to 100% of the results. So let's fast forward to the end of the analysis. DSS Studio will report results. Of course, there will be a short method section for you to make it easier. And you will output also other files, including the tractography of the positive correlation and also the negative correlation. Then check out the report in HTML format, or you can load it on a web browser. You see there's a method sections, and the results will be reported, say here, positive correlated with BMI. You will see the FDR value, see if the result is significant, and report the location and the precise location of where the tracks show correlated with BMI. Here's the negative correlation. We'll see where are the tracks are positively correlated and negative correlated with the BMI values. Also, we could see the result in 3D. Click here, step C3F. DSS Studio would open up tractography. We could see positive correlation, see where the tracks are. And those either here we could have negative correlation, see where those tracks are located. And we could change the color to assign the color. So negative correlation in blue, positive in red. So we could compare them together and then inspect the results. So on the left-hand side, you can see the slice locations. There's another way to present results in a mosaic view. So this would be nice for publication purpose. If you want to look at where those findings are located, switch on the right to the mosaic view, you will see how tracks are plotted across the T1 weighted image, and you help you to identify the anatomical structure and all the findings. Here is your summary. I would say tractometry, the conventional way, is still the first choice. If you can find a minimal finding, there's no point to go for more complicated methods. But in real life situation, a lot of time we may not have enough power. We may not be able to locate the precise location of the findings. And in this kind of a situation, you may consider differential tractography or correlational tractography to pinpoint where are the findings and also improve the significance. Thank you very much for your attention.
Hello everyone, my name is Thijs, and today I'll be trying something new. I'll be explaining you all about interpreting neuritor fiber density changes. So the emphasis really here is on density. And we'll find that this is quite a complicated thing, even though everybody does interpret density metrics all over the literature. So in my presentation, you'll often see these little figures like this one here and this one on the right here. And all you should know to understand what I'm talking about is that this will represent a voxel, and these little orange dots here, or other colored dots, they will be axons coming in and out of my screen. So this one's pretty densely packed, and this one would have a lower neuritor fiber density. Alright, so first, where do we even get density metrics from? Well, there's different techniques, Today we're of course talking about diffusion MRI, and there are several models that provide neurite density, fiber density, signal fractions, and the like. So there is NODI that you may know as a neurite density metric. There is the CHARMP model. It has an intra-axonal signal fraction, which is kind of similar. There's the apparent fiber density metric, sometimes called fiber density for short, many others. So they're all slightly different. The models have different assumptions. That doesn't really matter. In the end, they aim to estimate some kind of a density metric in each voxel of the axons. So the total amount of axonal matter, which is modulated by the count of axons in a voxel, as well as the individual axon volumes. So in this presentation, I'll be relying mostly on the apparent fiber density metric, and I'll just be calling it fiber density for short. But the conclusions towards the second half of this talk on how to interpret density metrics and all the problems that come with it, they apply broadly to all the models above and any density metric. All right, so apparent fiber density then is actually a quite interesting one, because we can get multiple apparent fiber densities even in a single voxel. We actually can get one per fiber population in a voxel. So for instance, here you got a template, and here you see the corticospinal tract, you see a little bit of the corpus callosum, and then you see the superior longitudinal fasciculus, and they all cross together in this voxel. With techniques like constrained spherical deconvolution, we can get a full fiber ODF that represents all of these. And the apparent fiber density then is the actual size of these lobes that belong to each of those bundles. So for instance, if you would have a control cohort and then you would have a patient group, you might find that maybe this tiny green lobe that actually relates to the SLF is decreased in the patients, and that would tell us that the fiber density is lower, specifically in that bundle, even though the other crossing bundles are not affected. So we can actually figure this out, which is much better than, for instance, the diffusion tensor imaging model, which in this case would actually show not a decrease in something, but actually an increase of fractional anisotropy. Due to the crossing fiber situation here, the anisotropy of the tensor actually increases even though something was damaged. So that is actually quite confusing to interpret, and we cannot even assign it to any of these three populations in particular. Hence why this one was developed. So how does that work then? Well, so we got these fiber ODFs all over the brain, and up front they are segmented in what we call pixels. So these little lines here, they are pixels, and a pixel stands for a fiber population in a voxel. So basically every one of these FOD lobes becomes one pixel here. And then the size of these lobes is added to these pixels as the metric, the pixel-wise fiber density. So here shown in the hot color scale, we can see that some pixels like here and maybe there, they have a high fiber density. Some of these have a little bit lower fiber density, which is not surprising because the density here is shared among different crossing populations, for instance. All right, how to analyze this then? Well, there's a framework for that, and it's called pixel-based analysis, and it's not that simple. There's like 20-something steps here, which I will now explain one by one. So no, let's not do that maybe. It's a little bit too complicated, it would take up way too much time. The gist you need to know about this is that essentially this is the same as voxel-based analysis. So like you know, voxel-based analysis, essentially you first create a population template, or maybe you use an atlas that comes with a template, but then you register all your subject images to this common template space. And in that template space, once everything's aligns voxel-wise, you sometimes smooth the images, you do your cluster stats or normal stats, and you get a p-value map. So that's the goal in the end. You get a p-value map and you can see which voxels, so which areas of the brain are significant to whatever you test for. So pixel-based analysis is essentially the same thing, but then rather than doing this for voxels, it does it for pixels. So in the end, you get a p-value map for each single pixel, you get a p-value. So doing that for pixels is the thing that makes the diagram on the left so complicated and adds so many extra steps. So you You can read all about why that is so complicated in the original work by David Raffelt and colleagues from 2017 in NeuroImage on the pixel-based analysis framework. Recently, with a number of colleagues, we've also written a review paper on this that hopefully explains some things in a way that is slightly more intuitive and helpful if you were confused before. And you can find the title here. Currently, it's a preprint that if you Google the title, you can probably find it easily. And hopefully by the time you see this video, or not shortly after, this should be published, hopefully in NeuroImage. Pixel-based analysis you can also do yourself on data, and that is awesome. The software you can find on the MRTrix website, www.mrtrix.org, and they also have a whole bunch of documentation on how you actually implement this in practice. So what do we get then out of a pixel-based analysis? So for instance, this could be one of those results. The thing that you're looking at here is a template to which, for instance, a number of populations, 2 populations maybe control population and maybe a patient population were registered, and we've done stats, we've compared them, and this comes out. So let's zoom in a little bit. Let's zoom in a little bit further on that region right there. So essentially what you're looking at is a p-value map that has a p-value per pixel. All the pixels, the little lines here that are black, the p-value is larger than 0.05, so they were not statistically significant. All the ones that are like dark red all the way to yellow and white, they are significant, with the white ones being the most significant, so very, very low p-values. So what's so cool about this? Well, you can see that in areas like here, so where, for instance, the corpus callosum, the little black lines here, and the cingulum, the white lines there, they cross. But in the statistical result, we found that the cingulum is significantly different maybe between the populations, whereas the corpus callosum is not. So we can actually have a pixel-specific result, and we can tell then, well, blah, 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 the cingulum was affected in this patient group, and so on, without needing to touch upon this other structure that was crossing here, but that was not significant. So we can do this for fiber density, because fiber density we get per pixel. And that's neat, but here's the bit where I tell you that fiber density is not everything that there is in this world. So let's say, for instance, you got 2 populations or 2 subjects or 2 things to compare, and one of them in a voxel or in many voxels looks like this, and the other one looks like this. So you're not mistaken. These 2 images are exactly the same, and the fiber density in both of those voxels is exactly the same. But as the title says, this is not everything, because these are only 2 tiny voxels. And of course, there is more data in an image than just 2 tiny voxels. There might, for instance, be more voxels. So the blue grid here represents voxels. And now we have here a grid 7 by 7, and there a different one. And what do we see? Well, within this bundle here, and within this bundle of axons there, the density is actually the same. If you look in each of these individual voxels locally, you can see similar densities of axons right there. So if in pixel-based analysis, or imagine voxel-based analysis, just the same thing, you register these to each other, the fiber density metric here, that is the same as there, will be mapped onto each other, and you will find that there is no difference in fiber density, because there isn't. But obviously there is a difference between these 2 bundles. This one here has way more axons than this one there. So fiber density tells us something about axons within a voxel, but not per se how many axons there are in the larger bundle. The size of these bundles tells us the second bit as well. And the size of the bundle, in terms of how wide it is, or in terms of how large the area of this bundle here is, and this area here, for instance, is larger, we call the fiber bundle cross-section. So it's a little bit like the volume, but only the volume along the width of the bundle, not along the length of it. And that's quite neat that we can do that, because that sets it apart from plain old voxel-based morphometry. So in voxel-based morphometry, let's say you got a patient group here and a control group right there, and the control group maybe has gray matter across a volume of a number of voxels, 2 in the simple example, whereas the patient group only has 1 voxel filled with some kind of gray matter. The densities are the same, but the volume is different, and voxel-based morphometry can pick this up. Fiber bundle cross-section is slightly more complicated, because let's say we got here the same situation compared to the gray matter with 2 voxels versus 1. Do we want to pick this up as a difference in fiber bundle cross-section? No, we don't, because this is a difference in length of this bundle. And because the length is different, it doesn't mean the number of axons or even the width or the cross-section indeed is different. The cross-section is the same. Whereas if they are pointed in a different orientation, then suddenly we do want to pick up this difference. And the pixel-based analysis framework will pick it up. So it will pick it up in this case, but it won't report the fiber bundle cross-section difference in this case. So that's a neat thing on the side. So now we're fully equipped. We got fiber density and we got fiber bundle cross-section, and we can do a bunch of things. So we can, for instance, know now that if this, let's say, is a control population bundle, that the fiber density could be decreased. And that could mean that in the patient population, we could imagine the bundle has indeed a lower density of exons, and in this case also less exons because of that. But another thing we've discovered is that we can measure fiber bundle cross-section. So we could also see a decrease in FC, fiber cross-section, and that's also a difference in number of exons here, but not by virtue of a decrease in density, but rather the cross-section of the bundle indeed. And then to complete this thing, like the title suggests, you can have both things changing as well. You could have a bundle where the cross-section is now less, but also the density is less. And to measure both of those effects and encapsulate them in one metric, we also have a metric called fiber bundle density and cross-section, which is computed as the product of fiber density and fiber cross-section. So both of those, if they have an effect, it will also end up in this one. And in this case, because both effects here effectively took place, you get double the effect. So the FDC is extra sensitive to both of these things together. All right, so practice time. I hope you've been paying attention and are also ready to reason a little bit further. We will just apply this the way you do. So how do you do this? You do research. You have 2 cohorts, let's say, controls and patients, and let's do a little cross-sectional analysis, right? So you set it up, you define your tests, you have your both populations, and you say, well, maybe the patients have a decreased amount of fiber density compared to the controls because the patients have some kind of damage in their brain. Makes sense. So you say, we're gonna test this statistically, and that's the thing you would write in your method section, right? So then you have your results section, and well, let's say we found that there was no statistically significant FD decrease. So that's what we found. So what's now the exercise? Because this is pretty straightforward, right? You've set up your stuff, results come out, and you just literally copy paste them and write about them in the results section. But then there is the discussion section, and now you're going to have to talk about it and interpret it. So I'll give you 2 options to think about a little bit here. What could you write? Well, here's option A. So remember, the result was there is no FD decrease. So let's say you write in your discussion section, patients show no reductions in axonal matter. So there was no damage in their bundles in the brain, and there was no degeneration, both of those types of language you might use in your discussion section. So not saying this is what you should write, but given these results, maybe this sounds like an option. Or what about this one? Patients show no microscopic reductions in exon density. So we're not saying then that overall they got less exons or whatnot. We just say we found no reductions in microscopic exon density because fiber density, so we stick close to it. So I'll give you a little bit time. If you're watching this beforehand, this might be a good point to pause the video and give it a bit of a thought Otherwise, think quick. All right. Well, so how do we tackle this? Well, let's just imagine a few situations. Why not? So could this be a situation that is compatible with the results? Well, let's look at it. No fiber density decrease. So we see here that the controls and the patients would actually look very similar. So the density indeed is not decreased in the patients. So this here is compatible with those findings. And it's also compatible with what we would have written in A here. So no reductions in axonal matter. No damage, no degeneration, no nothing. Sounds legit. But yeah, of course you've been paying attention, and I spoke to you about the fiber cross-section, which in this experiment we have not checked. And that's essentially the pitfall. We have not looked at FC. So this scenario may just as well be right, because indeed between these controls and these patients, there is no decrease, there is no difference in fiber density locally. So this also fits the results. But obviously here, you could not write point A there. There's obviously some damage or degeneration going on in the patients versus the controls. So how do you resolve this? Well, you should have just tested FC up front. If you had tested FC, then you could have distinguished this scenario from this other one and actually come up with one of them, and then you could sensibly write maybe A if you would have found this one. But now that we have not done these tests, the best we can write is just B, which, you know, if you read it, is literally what this here says in the results, which is pretty boring. So we have little to no margin for interpretation because we didn't test FC. All right, we're not going to fall for that again. So this time we do a longitudinal analysis, 2 time points, patients early after the diagnosis of some disease, and then longer term, those 2 time points we check. And we test whether the fiber density in the long term is decreased versus the fiber density early on. Maybe they degenerate over time. But now we're not going to get caught out, and we test the same thing for FC. All right, let's say we find that there is an FD decrease, but no FC decrease. So the density goes down, but the cross-section does not. So how do we interpret it now? Well, we could say the patients undergo reductions in axonal matter, right? Damage or degeneration because FD decrease. Or would there be another option? What do you think? Well, let's go again through some scenarios. So would this be an option? Early on versus longer term, fiber density decrease? Check, it is indeed decreased. No FC decrease? Indeed, the bundle is the same. So indeed, in this case, we could write A. There is obviously damage. But what about this scenario? Density decrease? Check. No fiber cross-section decrease? Well, check. It's not decreased. It's increased. So where did you get caught out here? You did not test for an FC increase. So what is this scenario? Does this make sense? Well, the number of exons here and here is the same. Well, let's say this was a disease that caused in the long-term inflammation that actually allowed extra fluids to develop in between the exons and push them apart. Does that make sense? Well, it could very well make sense. And you didn't get it. You haven't even thought about it because you didn't test for FC increases. So that's the pitfall. You need to test for FC increases. All right, we're not going to get caught out again. There's the last question. So 2 time points again. Same scenario, early after diagnosis and then longer term. We also test again whether long term versus early on the FD is decreased. We test whether the FC is decreased in the long term, and we also test whether it's increased because we got caught out before. But now we have a different result. We find no fiber density decrease. So density is the same, but we do find that the cross-section is decreased. How do we interpret it? Well, we could say, again, patients undergo reductions in axonal matter, damage or degeneration, maybe based on the FC decrease. Seems to make sense. But again, is there something else we could imagine? Think about it. Okay, let's again touch upon some scenarios. So how about this one? No FD decrease, check. Density here and there is the same. Fiber cross-section decrease, well, check. The bundle indeed did grow thinner. So that makes sense. But can you imagine something else? How about this one? Fiber density decrease? No, it did not decrease. It increased. Fiber cross-section decrease? Check. It did decrease. So we did also not check for a fiber density increase. But what the hell would a fiber density increase even be? What would it be? Well, again, the same number of exons are here, but they got closer to each other. So maybe this was a later stage of the disease already, and we took 2 time points later on, where there was already some damage here in density, but now the tissue collapsed into atrophy. And maybe that is what we picked up, the tissue collapsing, which in fact makes the fiber density potentially increase, but of course combined with a decrease of fiber cross-section, and that together makes the picture. So pitfall, we didn't check for increases of fiber density, so we should really check the whole bunch to get the full picture. That's the message here. Alright, so it's quite complicated, and I won't go through all the transitions here, but the gist is that indeed you do need to consider every possible arrow and every possible value. For instance, this might be from here to there a fiber density decrease, but what if it is followed up by a collapse? Then we end up here. And throughout the collapse, indeed, the fiber density might increase. All these things can happen. So you need to consider all the combinations. This can get even more complicated if you consider crossing fibers. I'm not going to discuss this right now, it's really complicated. But essentially, if you got crossing fibers and one of them, let's say the orange bundle, is damaged, it can actually start to affect the values of the other healthy bundle just as well, because it might collapse along with the whole structure. So not digging into that, all of that and many more examples are explained in our review paper. So do take a look there and give yourself some time to study and let this sink in. Okay, some conclusions then. Well, what do you know? Interpreting fiber density is pretty tough. That's what we figured out today, I guess. And there's many more questions that you should look into yourself, I think. But I hope this made you start to consider that maybe you need to think sometimes a little bit harder about what these things might mean. So what were the main pitfalls we discovered today? Well, ignoring the size of the structure, the fiber bundle cross-section we called it, that was one of the main pitfalls. So fiber density alone was not enough. We also need this other one to complete the picture. Not testing or considering opposite effects, so increases of both fiber density as well as cross-section, both do have sensible interpretations. If you don't check them, you'll never know that you might have discovered a case like those. Ignoring stuff and space between exons, so we saw a case of edema or maybe inflammation that makes a bundle swell, that happens between the exons, and maybe a bundle collapses. That also happens between. The exons are not affected, but the metrics are. So you need to be creative in your thinking. And finally, what I didn't even touch on today, an absence of statistical significance does not prove an absence of any effect. So basically, whenever I said we found no differences, well, actually we don't know. We didn't find any significant ones, but we might have just lacked statistical power. So actually that puts an extra unknown in some of the findings. So even when doing everything right and considering all the things, there's often still a range of explanations that are technically compatible with a given finding. And this just represents the limitations of the data and the methods, and you'll just have to deal with that. So ultimately, the only way beyond those limitations in specificity is to rely on assumptions, and that is not per se a bad thing. Oftentimes you have a sensible context. If you know in a disease that you can expect inflammation, then you might just as well hypothesize that and check that with your findings and draw conclusions based on that. Okay, I hope I gave you some stuff to think about. Enjoy the rest of the session.
Hi everyone, my name is Michal Ben-Shachar. Today I will be speaking about tractometry applications to high-level cognition. So our goal in this talk is to map between white matter pathways and cognitive functions. The main idea is to use tractography to identify known pathways in individual brains, extract microstructural properties of these tracts, and then examine associations with various cognitive measures of the same individuals. This could be done in the form of correlations with mean tract properties or along the tract, or it could take the form of group comparisons of mean tract properties or again along the tract. We can start this story with two early findings that relate white matter structure with reading and speech abilities. The first study demonstrated an association between anisotropy and reading scores in a sample of college students. Some of them were poor readers and some were good readers. Another study that you can see here, which was published around the same time, showed a significant group difference in anisotropy between adults who stutter and controls. So both of these studies made a large impact in their field and yielded multiple follow-up studies in adults and children. Also, both of them used voxel-based methods after registration of all the brains to a common template space. If we do that, we run the risk of sampling in a single template voxel different pathways for different participants. This is true particularly if there are differences between the groups in the volume of specific pathways. All right, when you see this symbol, this means it's your turn now. And if you're watching this task online, feel free to respond to the quiz questions in the chat window. Or if you're watching offline, you can pause here and then press play to see the answer. So the question here is, are there any other methodological concerns? So regarding the left graph, it is risky to calculate a correlation value on the entire sample when we have two subgroups that differ on both axes. As for the graph on the right, remember to pay attention to the values on the y-axis. If you get values that are near or under 0.2, we might be sampling gray matter, at least in one of the groups, or possibly in both groups. Actually, there are many voxels in which voxel-based comparisons have limited power, primarily because of individual differences in shape and volume. Some white matter structures, like the corpus callosum, are more susceptible to this problem, as we show in this power simulation. So the chances of finding an existing group difference in the corpus callosum using voxel-based methods are not too good. This is where tractometry becomes handy. In this early study, we used deterministic tractography to identify the corpus callosum in about 50 children, and then segmented it according to cortical projection zones in every individual child. What we do next is we chop these fibers near the mid-sagittal plane, where we know that the directional coherence is really high because all the tensors are oriented along the left-right axis. So now we have a segmentation of the mid-sagittal corpus callosum in each of these children. And if we do this on the entire sample, this allows us to identify comparable voxels in the callosum, regardless of shape and volume differences. So all of this hard work paid off when we identified a significant correlation between radial diffusivity and phonological awareness in these children. Importantly, this correlation was selective to the callosal fibers that connect between the left and right temporal lobes, which actually makes a lot of sense. And you can read the full story in the paper itself. Unfortunately, this trick of chopping the callosum and zooming in only on the mid-sagittal plane is rather unique to the corpus callosum. We can't really do that in other pathways like the arcuate fasciculus, which is a really interesting pathway to study in the context of speech and language and reading. And things get even more complicated because diffusivity parameters can vary a lot along the trajectory of the tract. For example, specifically in the frontotemporal fibers of the arcuate fasciculus that you can see here, you typically see a range of FA values that may vary all the way between 0.3 and 0.7. And this is a lot if you recall that the entire range of FA values in white matter is between 0.2 and 0.9. So we know why this happens in the arcuate, and it has to do with the divergence of parietal fibers that happens at the point of maximum curvature of the tract. But these fluctuations are not unique to the arcuate. So we have to find another solution to analyze associations in a manner sensitive to the variability along the tract. One solution that we used in this study is to extract diffusivity measures from a predefined segment, and then sliding that segment backwards and forwards to see how stable this correlation is. Using this method, we found an association between the frontoparietal SLF fibers and mental math calculations. There's a good reason why we analyze these frontoparietal connections, because fMRI studies of mental math consistently find activation in inferior frontal and in inferior parietal cortex. And in general, I'd say these methods are particularly appropriate for hypothesis-based studies. And this is not unique to our own research. Other groups have also used similar methods for dividing the tract into shorter segments and analyzing correlations with cognitive measures in each segment. For example, in this study by Ido Tavo and colleagues, they found a significant association with face recognition in the anterior right inferior longitudinal fasciculus, while scene recognition was associated with a posterior segment of the ILF bilaterally. In more recent studies, we calculate the correlations along the trajectory of the tract using AFQ. For example, here you can see associations with performance on letter and category-based fluency tests in multiple sclerosis patients. Letter-based fluency means that you're asked to say as many words as you can that start with a given letter in 1 minute. Category-based fluency is the same thing, but the words are produced based on a semantic category cue. Tractometry methods detect a significant association with letter-based fluency in the left frontotemporal arcuate. On the other hand, performance on category-based fluency tasks correlated with FA in the right IFOF. So this finding is one of many that support the dual stream model of dorsal and ventral pathways for speech processing. A few things to remember when we use these methods. Of course, we have to correct for multiple comparisons because we carry out about 100 statistical comparisons along the tract. Another thing is we always show scatter plots to assess the distribution of the individual values within the significant cluster. But it would be wrong to run statistics, again, only on the significant cluster. So don't do that. And one other thing is we typically calculate the correlations only between the two regions of interest that were used to identify the pathway, in order to avoid the parts that are closer to the cortex, because the tracts are less reliable and the variability is too high when we get closer to cortex. So here's another quiz for you. And this finding from a recent paper: is there any methodological issue we should be concerned about? And again, we should always look at the values on the axes. In this case, the FA values are around 0.1 to 0.13. So we might be worried about registration and perhaps partial volume sampling of the gray matter. So we've been working with tractometry methods for close to a decade now, and I think we reached the stage that we can trust these methods not only with standardized test scores, but also with really elaborate experimental measures of cognition and behavior. For example, you can have people walk on a split-belt treadmill and measure different parameters of locomotor adaptation. And it turns out that the magnitude of adaptation correlates with mean diffusivity in the inferior cerebellar peduncle. In another paper, we showed that your ability to synchronize finger taps to an auditory rhythm is associated with FA in the arcuate fasciculus. So I think there's a lot we can learn from using these methods with very specific cognitive measures. As another example, we have a series of papers that shows that sensitivity to word structure is associated with diffusivity in bilateral ventral pathways. For example, here we show that when you read in English, your sensitivity to word structure correlates with FA in the left and right IFOF. Personally, I think the most exciting aspect of these findings is that they generalize across English and Hebrew, two languages that have very different morphological systems. Of course, there are also many differences in the details of our findings from Hebrew and English, but the similarities are encouraging because they suggest that we're capturing something reproducible and something that has to do with abstract word structure beyond different scripts and morphological systems. We also show that the results generalize across modality and task and across different analysis pipelines, whether you're using deterministic tracking with tensor fitting or probabilistic tracking with constrained spherical deconvolution. So given how noisy these measurements are, such generalizations are really important. So this will be our last quiz, and feel free to write in the chat any concerns that you might have here. One issue here is that we have 4 data points for people who have a lateralized arcuate and 2 data points for those who show bilateral arcuate. It's not a scenario in which we want to calculate a correlation, not only because the sample size is too small, but also because data points are not well distributed along the x-axis. One of the things is if you calculate a Spearman's correlation, don't draw a line because Spearman estimates monotonic, but not necessarily linear relations. One last point I want to put out here is that tractometry methods also allow us to explore new frontiers, measure pathways that do not play a role in the classical models, and map associations between their properties and cognitive measures. 2 such pathways that I want to mention here are the frontal ascending tract and the vertical occipital fasciculus. We have tractometry results that show a group difference in the frontal ascending tract between adults who stutter and controls. In adults who stutter, we also found an association in the ascending between mean diffusivity and speech rate. Studies from Hiromasa Takemura and colleagues, among others, find associations in the vertical occipital fasciculus in relation to visual perceptual processing. So I think there's a lot for us to study, and we should allow ourselves to explore other pathways beyond the usual suspects using tractometry methods. To summarize, I argued that tractometry can be used to discover novel relations between white matter structure and cognitive function. I also want to highlight again the importance of generalizations and reproducible research in this field, not only across different languages and task modalities, but also across different research groups and analysis methods. I want to take this opportunity to mention a few points of caution concerning missing pathways and tracked endpoints. The first point is, if we didn't find a pathway, we didn't learn anything about this pathway. The second point is, strange things might happen in tractography when we get closer to cortex, though I'm not sure I would take the precise endpoints very seriously when we interpret our findings. And finally, the main cautionary note I want to leave you with is this. Finding a correlation doesn't indicate that we found the function of a given long-range pathway. The large white matter tracts that we can measure with diffusion MRI are very likely to be involved in many cognitive processes. The associations that we find may be task-dependent, modality-dependent, or context-dependent. So I think we should be careful in our interpretation and try to avoid assumptions about one-to-one mapping between tracts and cognitive functions. And with that, I'd like to thank the organizers for this great session, and many thanks to my current and past students, our collaborators, and funding agencies. And thank you for listening.
Hello everyone. So today we're going to be talking about multidimensional data analysis derived from tractometry. So as of now, you probably learned in the previous courses how to derive track profiles, for example. So here is just a brief recap of how do we extract these profiles. So first of all, we start with a whole brain tractogram for each of our subjects, and then we extract a set of tracks from that whole brain tractogram using various approaches. These could be automated techniques, for example, or via manual delineation. And then for each of these tracks, we define or we extract a center line or core streamline, which is going to be used later on to project different metrics. And this center line will, of course, vary for each tract. And then what we do is for each vertex along the tract, so each streamline is made of different vertex, we look at each vertex, which point of the core is the closest to it, and then we assign it to this core. So we create these kind of assignment maps or label maps. And then what we do is simply map a certain metric on the track and then take the average within each of these sections. This can be a weighted average, for example. So depending on how far you are from the core, you might give less value to the metric. And if you're close to the core, you give a bit more value. And then you end up with these track profiles, basically, for each of your tracks and each of your subjects. But the tractometry philosophy is actually not necessarily based on track profiles. It's more of a general framework, which was introduced in ISMRAM 2011 by Sonja Bells, where, as they say, it's try to combine multiparametric data along multiple tracks. So in their abstract, and actually in the paper from Silvia De Santis, they used tracked averages along multiple tracks, but they mapped various MR-derived metrics. So in this talk, we're going to be looking at tracked averages of multiple tracks in the first part, and then looking at track profiles a bit later on. And we're going to be talking about two concepts. So first, the dimensionality reduction concept, where we have a high dimensional space, such as having multiple tracks and multiple metrics per tract, and try to reduce the space into a low dimensional space, which is a bit more interpretable, hopefully. And the second concept we're going to be looking at is the idea of anomaly detection, where we try to look for outliers in our data. So dimensionality reduction. So typically when we work with multiple diffusion MRI measures in a univariate manner, we often have this statistical problem where each of these metrics are treated independently and tested against our hypothesis. And then we have this multiple comparison problem. The other problem by treating each of these metrics independently is that we totally ignore the relationship between these metrics. So they might actually be probing complementary information. So it would be nice to have a framework that could combine all of these metrics into some meaningful components. And that's what we're going to be doing today. We're going to be replicating a figure from one of our recent paper where we leveraged dimensionality reduction to create this new subspace where we projected different tracks and different metrics. And this was done in a cohort of typically developing children. And the nice thing is that these components, these new components, actually correlated better with age, so with the developmental age of these subjects, rather than any of the individual univariate metrics that form these components. Okay, welcome back. So we are now in the Jupyter Lab environment, and we're going to be replicating an analysis from this first paper here about dimensionality reduction of diffusion MRI metrics. So the goal here is as we will see, we will have a set of bundles with a lot of diffusion metrics that are mapped on these bundles. And we want to reduce the space of metrics. So basically, as you see here, we have 10 metrics, and we want to reduce this down so that we can interpret the results a bit better. So first thing is we're going to be loading our libraries. These are mostly Python libraries for plotting and for machine learning. And let's load our dataset. So as you see, the dataset has 792 inputs and 10 columns. So let's look at these columns. We have an ID column, a bundle column. So this states which bundle we're looking at. And then these are some features for this bundle. So the mean FA, the mean AD, RD, mean diffusivity, etc. So these are DTI metrics, and these are more advanced metrics derived from Hardy. So AFD is the apparent fiber density, NUFO is the number of fiber orientations in the bundle, AFD dot is a generalization of AFD, and FR is a restricted signal fraction derived from charmed. So as you can see, all these metrics may have some things in common, and some might pick up different part of the signal rather than others, but these are all features. And if you look at the data, here's what I was meaning. So basically we have our subject's ID, the bundle associated to each subject, and then the mean of each of these metrics. And this is repeating. So as we go down, the same subjects will reappear with different bundles. So for the ease of simplicity, we're going to select only a subset of these bundles for now so that we can better understand what's happening in the background, and also for visualization. So let's pick like some main bundles like the corticospinal tract, the uncinate fasciculus, the arcuate fasciculus, the splenium and the genu of the corpus callosum, and the optic radiation. And I'm taking only the ones from the right side for now. So we have now this data set, and we drop the ID and the bundle features because we're going to be using only the macrostructural metrics. So we end up with 216 rows and 8 columns. First, we can look at the correlation between the features. So basically to see if there's any link between any of these metrics. And as you can see, we see some correlations between different metrics as we expect. For example, FA is highly correlated with AFD because these two are probing the anisotropy, let's say, in the tissue. And similarly, FA is also correlated with FR. This is also an anisotropy metric. So you can kind of see already, have an overview of different relationships between all of these metrics. So first, because all of these have different scales, so MD has a really low scale or values compared to FA. So we want to scale everything to the same order of magnitude. So this is done using the standard scaler and scikit-learn. And we're ready to go. So first, let's look at the principal component analysis. So we're going to specify 2 components. So we want to reduce these 8 metrics to 2 major components. And that's it. That's pretty simple. But now we're going to have a look at what's happening in the background. So by looking at the explained variance attribute, we can see that the first component of the PC explains 48% of the variance in our data, and the second component explains 35% of the variance. So that's already a good indication that selecting two components accounts for almost 85% of the variance. And we can also look at which of our features contributed to each of these components. So you can see here I have 8 features and here I have 8 rows. So this is for component 1 and this is for component 2. So we can see that for component 1, we have metrics that contribute the most to this component related to FA, so the first one here, AD, RD, AFD, and FR, also AFD total. So these values are basically all the anisotropy measures. So we could call this component the tissue restriction or hindrance component. And the second one has mostly inputs from AD, mean diffusivity, and NUFO, so number of fiber orientations. So this PC2 is kind of different from PC1 in the sense that it could be named as the tissue complexity component, if you try to interpret a bit what they mean, let's say. So now we're going to apply this transformation to our whole data and look at this new subspace that we created. So the PC1 versus PC2 for each of the subset bundles that we've selected at the beginning. So first off, each point in each cluster represents one subject. And each cluster represents a tract, as you can see here by the color map. So if we try to interpret how these tracks fall in different portions of this new space, you can see that they cluster pretty well at different parts of the space. And remember, PC2 was the component about tissue complexity. So if you look at the top of PC2, you see tracks that are more complex, like the arcuate fasciculus, which has a lot of crossings with other fibers, than tracks located at the bottom of the graph. For example, the uncinate fasciculus. This is a single fiber population bundle, or the GNU and the splenium. So it's kind of intuitive to interpret how these tracks fall in the space. But the nice thing about this space now is that for each cluster, one could, for example, compute the mean of the cluster and then compare each point to the mean by computing the Mahalanobis distance. And then this gives you an individual distance for each of these subjects to see how this person here, the splenium of this person compares to the mean splenium of all of the group in this new feature space that takes into account all of the different metrics. So that's a way of doing single subject analysis using multidimensional data. Now we're going to be looking at another way of doing it, which is with deep autoencoders. And we're going to be looking at two different autoencoders. So let's scale the data now using a min-max scaler, which is preferred for autoencoders. So again, we have our 216 inputs and 8 rows. So the idea behind an autoencoder is to simply take the data in, compress it into a latent representation, and then decompress it and compare the output with the input. And this should be the same. So basically by this compression, we're trying to learn only the important features in the data. And then if we specify that we want to encode it with 2 neurons, so in the middle layer, that means it's basically the same principle as with PCA, that we want to extract two components. And with every neural network, you can specify activation functions. In this case, if we specify linear activation functions, this is going to be replicating basically what we do with a principal component analysis. So let's first train our model and then fit the data and see that as we train, the error goes down. So that's what we want. We want to see this going down. And let's train quickly a second autoencoder, but this time using nonlinear activation functions. And then we're going to be comparing the output of both with PCA. So let's plot the previously generated graph, and then the autoencoder 1 and 2 features for both linear and nonlinear. And then as you can see, each of them captures different information and data. So it's up to you then to see which technique you want to be using. But here are some pros and cons. So for PCA, of course, it's pretty fast. There's no parameter tuning. And of course, the problem is that it is linear. You can only model linear relationships between the features. Whereas with autoencoders, you can try to see if there is non-linearities between each of these features. But it requires more tuning. Finally, another way to do dimensionality reduction, which is pretty popular these days, is looking at UMAP. Fortunately, this is pretty easy again to implement. The thing with UMAP, however, is that it is mostly for visualization. So it is good to see different clusters, as we can see here. It's pretty nice, nicely separated. But the distances in this space or the organization doesn't mean much. So we have to be careful here with trying to do more quantitative analysis of these different points. It is a bit slower than PC also, but it scales pretty well. The more data you have, you're going to see it works pretty good. And again here, at least from the interpretation side, you can see that each of these clusters represent different tracks and they are nicely separated. Keep in mind here, we didn't input the tract label in the training. This is purely based on microstructural metrics for each of these tracks. Okay, so for the last portion of the talk, we're quickly going to go over anomaly detection. So before understanding anomaly detection, there's kind of a bigger overarching framework, which is called normative modeling, which is really trendy these days in the field, where basically you try to compare one subject against a group of healthy controls and try to see, is this subject part of the same distribution of the group, or how far is this subject deviating from the norm, for example. And this can actually be done with track profiles quite naively, because imagine you sample a group of healthy controls. Let's say you have 100 subjects, and you look at the mean apparent fiber density along the corpus callosum, and then you sample at 20 points, and you plot each of these subjects, so each of these purple lines here, as a normative distribution. And then a new subject comes in with a certain brain condition, and you do the same analysis. So you plot the points along the tract, and then you project it on this new space. And then you look, is this subject falling inside some sort of interval? So for example, 1 or 2 standard deviation from the mean. Or is this subject actually deviating from this? And this has been proposed and done a couple of times in the past. But it's actually quite an easy way to compare each point along the tract to see which points are kind of inside and outside this confidence interval. So that's called the z-score approach. Now, as you see here, all these points forming this bundle. However, anomaly detection can also be done in a more multivariate approach. So a bit like we've seen before, if you were to project all of these points into a subspace, you could see how far is one subject from the rest using this Mahalanobis distance, which kind of takes into account more than a simple or single track profile and one measure for one tract. But we're going to be quickly going over another technique, which is based on deep learning in the next slide. So a way of thinking about normative modeling could also be to train a network using only healthy controls data to learn what would be a healthy brain, basically. So if you give as input a set of track profiles which only come from healthy controls, and in this case we use the deep autoencoder. So we compress the data and decompress the data, and the goal is to obtain the same input in the output. And you do this and you learn basically what latent representation best represent this healthy control database. And then you give in a new subject, so one of your patients, for example, into this network, and you ask, what's the reconstruction error? So what's the output minus the input? And it might actually fall outside of the distribution if this subject has anomalies that the network has never seen before. Or it may actually fall within the distribution if the subject is simply as normal as the rest. So if you want to know more about all of these frameworks, there's quite a lot of them and a nice community around them. So there's the whole AFQ suite that has pretty good tools. I would also point you towards DiPy if you want to work with Python code and diffusion in general. And there's also TractSeg, which is quite useful to derive these tracks. So with this, I would like to thank you for your attention.
Everyone, my name is Sila, and today I'm going to be talking about applications of tractometry to brain development. I'm based at the Cardiff University Brain Research Imaging Centre, and let's get started. So just to give a little bit of background on brain development and why that's interesting, particularly when looking at white matter microstructure, we know that the brain dynamically develops over childhood, and by about age 5, the brain size is approximately 90% of its adult size, but the contributions of grey and white matter vary with age. And MRI has taught us a lot about what happens with grey matter and white matter macrostructure with age. So we know grey matter volume decreases into adolescence and white matter volume increases into early adulthood. And this really interesting period occurs in childhood brain development where the biological process of puberty results in circulating hormones in the brain, and that can influence the way that microstructure develops, particularly given children have different ages of pubertal onset and hormonal changes. So age-related development has been well characterized in DTI studies. We know the microstructure gets more organized with age, and patterns of development can differ between different fiber pathways. So for example, here, the two examples here are the inferior longitudinal and superior longitudinal fasciculus, and the rate of change can differ between tracts. Obviously, you probably all know why white matter is important, particularly given the talks that were presented today. But because these are the fiber pathways connecting different brain regions and they're dynamically maturing in childhood, all these different elements of the microstructure can affect the way information is transferred across the brain. So the myelin thickness, the axon density and diameter. So the intraaxonal compartment, so like the density of the intraaxonal compartment and the density of the total axon, including the myelin. These can all influence the way information is transferred, and we know bigger axons, and more myelinated axons conduct faster. So advances in diffusion MRI techniques, some of which you would have heard about today, allow us to tease apart these aspects of the microstructure. So just to give you a brief outline of what I'm going to discuss today, the first part I'll be talking about two separate applications of tract-based analyses. So the first is talking through a longitudinal pixel-based analysis that we did a few years ago, and more recently tract-specific development of white matter microstructure and how that depends on your diffusion MRI acquisition. And then in the second half of the talk, I'm going to talk a little bit about along track profiling, and you would have heard a lot about that today, so I won't go over the background of along track profiling, but more so do some interactive plotting using multi-cohort data, which we have combined 4 different cohorts to look at longitudinal changes over childhood, adolescence, and early adulthood. And show some demonstrations of how to visualize your data. So to start off with, you would have heard earlier, pixel-based analysis is one of these recent advances in diffusion MRI analysis techniques that allow us to gain both sensitivity and specificity to microstructural attributes. This is implemented in MRTrix 3 software and aims to overcome a lot of the limitations with diffusion tensor imaging, as rather than taking some average value over a voxel,
we can get fiber-specific information, and particularly when we're performing group comparisons, we can look at specific fiber pathways that may be changing between groups or even along dimensional variables or continuous variables. Now, the main measure of interest here is the apparent fiber density, or the AFD, and this is proportional to the interaxonal volume fraction, meaning all of these compartments added together allow the inference of apparent fiber density. And as I mentioned, group-wise statistics can be performed to isolate specific fibers that might change with your variable of interest. So in the context of neurodevelopment, longitudinal studies are scarce, particularly those that apply advanced diffusion imaging analyses. So our aim was to investigate longitudinal fiber development using pixel-based analysis in a cohort of children aged 9 to 13, followed up between 16 months apart. So the most the interesting thing that we were looking at, or what we were interested in, was which regions were dynamically developing during pubertal onset, and whether these affect the rate of fiber development. So I won't go into this in detail, but we went through a recommended pre-processing and analysis framework and computed multiple fiber metrics, including fiber density, fiber cross-section, and fiber density in cross-section, calculated difference maps, and divided that by the time interval to make sure we were sensitive to the change per unit time, and perform whole brain statistics with reference to change in time, sex differences, and pubertal interactions. Now, our main results showed that there were particular regions of the brain that were significantly changing or increasing in fiber density over this particular period. And this is a dynamic period of change during pubertal onset. And this visualization just shows how in these specific tracts there are significant increases in fiber density. For example, here 3.5% increase, and here 2.7% increase. And there were other regions like the corticospinal tract and the cingulum implicated as well. So these sorts of analyses were initially applied to single shell data and recommended to use high B values. So in the previous example, we had a B value of 2800. But increasingly studies are adopting multi-shell acquisition. So our next question really was, how do we tackle this age of big data and lots of acquisitions, particularly lots of diffusion MRI shells, and what is the best combination of B values for this sort of pixel-based analysis, and does this affect the estimation of AFD? So in this example, I'm going to talk about tract-specific development of white matter microstructure and the dependence on your diffusion acquisition. So in this scenario, we wanted to, answer the question of whether more diffusion MRI shells were better for estimating microstructure given you have more information, whether this influences pixel-based analyses, and whether this allows us to gain or lose sensitivity to age relationships. And recent advances in MR gradient hardware allow us to get these more detailed microstructural measurements. So we aim to measure the impact of different B-value sampling schemes on developmental patterns of AFD. In this particular group, this was acquired at Kubrick. 78 typically developing children were scanned, and we acquired this multi-shell diffusion acquisition with B-values ranging from 0 up to 6,000. And you can see here in this image that the higher you go in terms of B-values, so this is 6,000, the greater specificity you have to the interaxonal water. So any inferences you have on the microstructure should be more accurate at the higher B-values. Just briefly on the pipeline, it was pretty much the same that you saw before. We take the diffusion MRI data, pre-process it, and then we map that to a fiber-specific template, and that's in population space. And then we used automated tract extraction using Tractseg to isolate multiple tracts of interest, and they're all listed here. These are the ones that we extracted in this particular study, and out of the library of 72 tracts, we chose approximately 30 of them so that we could kind of hone in our analyses on developmentally sensitive regions. So just to briefly summarize the results, we took the AFD along each fiber pathway at each B value. So this is 1200, 2400, 4000, and this last column is 6000. And then performed hierarchical clustering to see if there were patterns of age-related development for each of the B values along each of the tracts. So just to orient you on this color scale, the R squared is the strength of the relationship between age and AFD at each of the B values in each of the tracts. So as we can kind of see here, age relationships were best described at moderate to high B values, as this many more tracts were significantly associated, and the R squared was much higher in these higher B value acquisitions. So we also noticed that most of these tracts were association tracts, and there wasn't too much of a difference between the B4000 and the B6000 in terms of added value. So just to zoom in a little bit, this top quadrant, you can see that everything is significant, denoted by these stars, and all of these tracts are significantly developing with age. But when we go down to these other two subclusters here, there are some tracts that are not very well characterized at these lower B values, and they're not sensitive to age relationships compared to at higher B values. For example, the uncinate fasciculus, portions of the first segment of the SLF, as well as the optic radiation. And just to visualize further, you can see in a tract where each of the B values provides sensitivity to age, these relationships are quite strong, but they do get stronger with higher B values. But then when we move to other tracts like the right SLF1, there really is a weak relationship with age at the lower B values. So when we apply this to the pixel-based analysis framework, we also observed that the overlap between results of the two higher B values or even the moderate B value were generally the same. But when you go to really low B values, the only real spatial overlap we observed was in the corpus callosum. So just to summarize this particular analysis, the developmental patterns that we observed were in line with previous findings, suggesting that the tracts that are associated with age-related development are what we actually expect in terms of what's known from DTI. And at higher B values, we're gaining more sensitivity to age relationships as AFD better reflects age-related differences in axonal microstructure. And that, in turn, helps us improve the characterization of axonal properties. Now, these relationships do differ across the brain. We observed a greater B-value dependence in left lateralized association tracts. And overall, estimating AFD in a tract-specific fashion in terms of age-related development is better characterized at high diffusion weightings as we are improving our correspondence with interaxonal properties. So that particular example, again, just highlights the importance of looking at things multidimensionally in terms of the acquisition parameters that we're interested in and the tracts of interest. Okay, so in this second half of the talk, I'm going to briefly touch on along track profiling and then spend a lot of time doing some interactive plotting using multi-cohort data. So again, what we know in terms of neurodevelopment is that white matter microstructure is driven by thickening of myelin and increasing axon density, but these properties can vary along the length of fiber pathways. So rather than averaging in one fiber like we did before in those specific tracts, we can segment tracts into different points and look at differences along the points as well as between groups. And this allows us to study subtle differences. And this is termed tract profiling, which you would have seen some examples of earlier. So our main interest in this particular application was whether these patterns vary in aging and development, and are there points along the tract that develop at different rates. So to show this example, we took some really rich data from 4 different cohorts, and our main goal was to leverage the power of longitudinal data. So all of these cohorts have at least 3 time points. Some of them you can see here, this Calgary preschool data set has up to 20, some children were scanned 20 times. So this is really rich data. And our age span goes from 2 up to 34. And you can see we've got a large number of scans with actually a large number of participants that were scanned multiple times. And we estimated fractional anisotropy, or FA, along 13 tracts and did that in 20 segments. So just to show you a brief example of one of the data sets, this is the Calgary preschool MRI data set. Some participants had multiple time points, some only had a couple. And then, as I mentioned, we did a long tract profiling using TractSeg and mapped diffusion tensor metrics onto these tracts. So in all of these data that I'll be presenting, these were acquired at lower B values, so we could only really reliably obtain estimates of diffusion tensor metrics. So that's why I'll be focusing on the FA results here. And just to give you an example of a few tracts and how we map the points, basically for each of the tracts extracted from TractSeg, they're mapped into 20 points along the length of the tract, and point 1 is anterior and point 20 is posterior. Okay, now I'm going to go through interactively plotting your data, more so looking at FA values along these tract profiles that we mentioned earlier. And I'm going to start off with the Calgary dataset. So this is all done in RStudio, and this is just my preferred way of doing it. And I'm just going to start by loading the packages that I need. The main thing that we're using is ggplot. And so just to briefly mention, the way we're going to visualize these data is by grouping the kids up into age 2 to 4 years and 5 to 8 years, and joining the demographics with the rest of the diffusion MRI data, which I'll run through in a second. But I'm just going to go ahead and run these plots in the background before I go ahead and explain how to get them. So they'll just sit there in the background. So basically the ggplot function allows you to input a data frame and maps aesthetics as to the X and Y axis. So for example here we're going to have a point along the X axis and FA of this tract along the Y axis. And I'm grouping these by the age group that we've defined earlier, and then smoothing the line using a GAM. And you can modify some parameters here to change the theme as well as the colors. And I've gridded now all of these tracts together so we can visualize them all in one. Just a reminder that FA value is on the Y axis. So if we just zoom in for a second, we'll be able to view the plots a little bit more clearly and talk about them in a little bit more detail. So just to orient you, the younger group is this color at the bottom, and the older group is the blue. And you can see in general for most of the tracts, the older group FA values are much higher along each point of that tract. And we can see that for multiple tracts. And this is what is expected, particularly for this age range where we're seeing large differences. There are some tracts where there might be some points that are closer together than others. For example, this point of the uncinate as well as the posterior portion of the SLF3. So it's a nice way to visualize your data to make sure that it's looking the way that you expect it to look. So another way that we can look at these sorts of data is by faceting across different groups. So here we've already split the curves up into 2 groups. If we want to split that up even further, we can facet them into multiple groups. For example, looking at sex differences. So that's what I'm going to do right here. I've already run this code, so I'm just going to bring up the plots now. So we're grouping by sex and then faceting by the age group. So you'll be able to see the 2 curves separately for each sex, and then grouped by age group. So the colors will be mapped differently this time. But the reason this is interesting to look at is that you might have age by sex interactions that actually cloud the picture. So for example, here we can see that 0 is females. There is a higher FA in females than males for the older age group compared to the younger age group for a lot of these tracts. So the arcuate fasciculus, the right cingulum, the left IFOF, and the left SLF. And this kind of gives us an idea of what is happening in terms of sex differences around the age of pubertal onset, because pubertal hormones start to rise around age 6 to 8 years. So this might be some sort of clue as to what's happening really early on in the piece. If I just pop the rest of the tracks down, we'll be able to see a bigger picture. And again, this just helps us get, when we're looking at neurodevelopment, it's not just a linear development. There's all sorts of things interacting, particularly when looking at difference between males and females. So similar to before, you can see the females are a little bit ahead in their development in SLF2 as well as the ILF. So that's just one example of how to visualize these sorts of data. So that's for the Calgary data set. So what I'm going to do now is bring up the NICAP data. So this is the data that I completed my PhD on. And this group of children were typically developing age 9 to 15. And they were kind of age matched for pubertal stage. So there are some children that start puberty earlier than others, but have the exact same age. And we can kind of see that in the figures when they do come up. So I'm going to go ahead and plot these data again, as we did before, just so we can see the big picture. And then because the age groups are so close together, we're probably not expecting too much of a difference in terms of age-related development in this cohort. But the one thing that we do see is some counterintuitive relationships, which again can be explained by puberty. And I'll go into a little bit more detail in a minute. So in this particular example, the FA again is on the y-axis and the point is along the x-axis. And we're grouping by those 2 age groups that I mentioned earlier. And soon we'll be able to see how actually the younger age group have higher FA in a few tracts compared to the older age group. So again here the orange color is the younger age group and the blue is the older. So for a few of these tracts, we've got higher FA in the younger age group compared to the older, which is counterintuitive. So here SLF3, left uncinate, as well as the splenium of the corpus callosum. So if we go ahead and do what we did before and facet these figures by sex, we'll be able to see what is going on a little bit further. So for example, in the left IFOF, when we split those data points up into males and females, you can see exactly what we saw with the Calgary data set, but kind of inverse, because this younger group is around the age of pubertal onset, and here we're a bit older. So the females are ahead in their white matter development compared to males at the younger age, so around the time of pubertal onset. So that's for the left IFOF. If we have a look at the other tract that was of interest, that's the left ILF. We see something very similar, and that's kind of along the whole length of the tract, more so in the midsection. We'll go ahead and look at the splenium, because this is a region that is known to be affected by pubertal onset. Here we've got, yeah, again, very similar. The females are a bit ahead most of the way through. And then finally, it was the left uncinate fasciculus. So this one is a little bit different, where we can see actually in the older age group, the males have higher FA, particularly in the midsection. So that, I guess, stresses the importance of visualizing your data, particularly when it comes to multidimensional data like this, where we've got multiple track points, multiple tracts, sometimes multiple measures. Here I'm just looking at FA. We could be looking at a variety of different diffusion MRI metrics. And it's really important that we try and tease apart these interactions, particularly neurodevelopment. Now, I'm just going to briefly mention the Leiden data as well as the Pittsburgh data, because these data sets have much larger age ranges than the other two. So these age groups here are 8 to 12, 13 to 18, and 19 to 28. And with these data, they kind of follow the expected pattern of increases with age as you get into adulthood. So I'll just show maybe one example just so we don't spend too much time on it. So just for a smaller number of tracts. And for these three separate age groups, the color gradient is similar to what we used before, where we're seeing the lighter colors are the younger age groups and the darker are the older. So in this data set, as I mentioned earlier, this is quite a large, rich data set of a few hundred scans. And because we're using longitudinal data, that really adds power to the conclusions that we're drawing. And you can see in this figure here, I'm just going to focus on the couple that we're seeing the expected patterns. For example, in the left arcuate there are some regions of the tract where there is this somewhat linear increase in FA between the age groups, but there are some regions that don't change much at all over age. Similarly with other tracts like the left CST, there's not much change with age, maybe something slight along certain points. Whereas there are other tracts, again, like the splenium that we saw earlier, that there is quite a large jump between at least the first and the second age group, which again might be due to puberty. And then is a significant increase forward, but not as much as we see earlier in childhood. So I've just focused on the left side of the tracts here, just so we're not bombarded with information. And just to finally show an example of the Pittsburgh data. So this is very similar to the Leiden data, except there's a few more time points, and we've got more data from the older age ranges. So I'll go ahead and plot the left side again for this cohort so that we can see these sorts of plots with more age groups. So we've got 5 groups here. And again, this is for the fractional and isotropy data. So we can visualize how these FA maps are changing with age. So you can see in this figure that it is quite nice to visualize these sorts of data with larger age groups, because particularly when there's this gradient of change. But what we can notice here along a lot of the tracts is they don't always follow the same shape with age. So, for example, there are some tracts like the splenium and the CST that the shape is quite overlapping. It does traverse upwards with age, suggesting that the age effects are there for FA. But there are some tracts like the SLF3 and the uncinate where the shape is quite different for the younger participants than the older. So that suggests that there's some tract point interactions where certain tract points are developing at different rates than others. For example, these mid-curving sections of the SLF3 and the anterior section of the uncinate fasciculus compared to the posterior. So that's another way of visualizing data is by increasing the number of groups that you're interested in. So just to take a minute to summarize, I guess at the start we talked a little bit about voxel-based versus pixel-based analyses and how pixel-based analyses can improve what we know with DTI studies, and then introducing tract-based analyses, which has been around for a while, but now with these more automated tract extraction techniques and tract profiling techniques, this can be more accessible for really large cohort studies in this age of big data. And then we spoke about longitudinal data and how that's really powerful for tracking neurodevelopment, particularly when looking at individual tract profiles and points along each tract. However, you know, one of the caveats of legacy data is that it's quite acquired many years ago over a number of years. So the conclusions that we spoke about earlier in terms of acquisition parameters and how we want to optimize these for really looking at axonal microstructure don't always apply in reality, unfortunately, because the data that is acquired in the past is more so suitable for DTI. But you can see that with the data that I plotted in the tract profiles, we are observing the expected profiles with age. So that's really promising that even with DTI data and looking at FA, we can be sensitive to those age effects. But obviously, future studies can attempt to tweak acquisition parameters to improve the correspondence to the axonal microstructure. And finally, I am a big fan of plotting data, and I hope that I've shown that can be valuable, particularly with my favorite is ggplot, but obviously everyone has their own interests. So I would just encourage you to plot your data and make sure it makes sense before arriving to any of your conclusions. So I'd just like to finish by acknowledging the organisers of the course, Catherine and Ariel, as well as my collaborators that were involved in the 4 cohort study, as that is new work that hasn't been published. And thank you all for your attention, and I hope to talk to you in the Q&A.